This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1109, recorded on April 26, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. Looking at the window, one would assume that since the sun is shining and the clouds are all puffy, that it's a beautiful day out there, but it's a little chilly and it's a little windy. It's not a perfect day, but I think I was told by the weather report yesterday that Sunday will be spectacular.
Here in Chelsea, it's 15 degrees Celsius and sunny. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it's 58 Fahrenheit, 14 C, and we have completely clear skies here. It's beautiful.
Yep.
We're expecting Brienne to join us, so she will pop in at any moment.
Vincent, you should have said it says 14 degrees Celsius.
Celsius. That's very good. Sometimes you're funny, Dixon.
Inadvertent, trust me.
Try the veal. I'm here all week. If you enjoy these programs, these science programs, it's W.C. Fields. We would love to have your support. Yes, because we give you science as it is and not fake science. So if you like that, go to microbe.tv/contribute so we can reach more and more people. A few public service announcements. PSA for ASV. Yeah, that's today. If you're interested in plant virology, there is a special plant virology lunch to sign up for at registration. If you didn't sign up, you can change your registration by following the instructions from your registration receipt. I tried to do that. I couldn't figure it out, but maybe you'll have better luck.
Will they be serving an all-meat meal at the plant virology lunch?
Right, all plants. They won't eat plants because they like them, right?
Right.
You can also change your registration and consider registering for a satellite meeting. If you register for any one, you can go to any of the 5. These happen the day before. The day of the meeting begins in the evening with a reception and a keynote, and that morning are the satellites. So there's invertebrate virology, a veterinary viropalooza, studying bats using a One Health lens, virology in a global society, and navigating virology funding opportunities with NIH, NSF, and USD. And we have another conference, Pandemic Preparedness Achievements, Current Challenges and New Frontiers. This will be 11 to 13 November in Trieste, Italy. An overview of known and emerging viruses, epidemic risk, networking, and sharing of information. So there's a website that we will put in the show notes that you can click on to get more information. And here's a new conference that I recently heard about. And, I heard about this at the Ian Lipkin symposium a couple of weeks ago. I sat next to the organizer and I said, you ought to have a TWiV at this conference. He said, yeah, that's a good idea. Can you advertise it? I said, sure, that's a good trade-off. So this is called Molecular Determinants of Zoonotic Viruses and Beyond. This is going to happen in March of 2025 in Freiburg, Germany, at the university. And this it's an Institute of Virology there in Freiburg. So that's next year. And things like emerged zoonotic viruses, virus-host interactions, molecular host determinants, structural biology and microscopy, novel vaccine strategies, therapeutic intervention. Let's see, some conserved speakers.
Conserved speakers, right.
All right, Brienne just texted me. She's not coming today.
Oh.
All right, but she was on the schedule, so we will just proceed without her.
We'll just have to gut it out for the 3 of us.
Jean-Laurent Casanova. I had my eye on him for a TWiV. Adolfo Garcia Sastre, Johan Yates, Gustavo Palacios, Tony Schounts, Silke Sterz. Now, Silke Sterz figures in one of today's paper. Silke is in Zurich, and I had her on a TWiV there a couple of years ago. She discovered the MHC molecule as a receptor for bat influenza viruses. Silke Sterz. Isn't that a cool name, Silke?
Yes.
I like different names, not Alan and Vincent, and Dick. Well, Dixon is pretty different.
Dixon is pretty good.
That's good, but Silke, it's so cool. Name some other countries that can be so cool. I really like it. So there'll be a link for that in the show notes as well. And don't forget to buy Dixon's book, The New City. You can go to DavePalmier.com. He also has a blog post about it that we will link to so you can learn more information about it.
Thank you, Vincent.
Now, we have some, what is it called? In the News, courtesy of Amy Rosenfeld. And we have an article by in the New York Times. It's an opinion by Zeynep Tufekci. That looks like a Turkish name, doesn't it?
Could be.
It's an opinion columnist, actually. And actually, she talks with Rick Bright. And so the problem is the headline, "This may be our last chance to halt bird flu in humans, and we are blowing it." This is just, what do you call it, Alan?
Hyperbole?
Hyperbole, clickbait, right?
Clickbait. Yeah.
Come on. What do you mean halt in humans? It isn't doing anything in humans, not spreading. Anyways, a nice picture of a cow. So Rick Bright is an expert on H5N1. And there's a fine line between one person and 10. By the time we've detected 10, it's probably too late. Okay. Well, You know what, Rick? What do you want us to do? We have antivirals. We have vaccines. Would you like us to cull all the birds on Earth? Anyway, that you should go read it if you can get into the Times. It's good to hear other people's opinions. Then we have sequences of, I can't get to this. Here we go. USDA publishes sequences of H5N1 on a publicly available site, 239 sequences from the U.S. clade that is currently circulating. And what I think is interesting is that these are still, they don't have any human signatures yet. Right? Human influenza viruses have certain signatures, which you'll actually hear about in one of our papers. And these don't have it. It's clear that all the cow viruses are from, they have a similar origin. Marco Warby has said this, he thinks that the virus may have been circulating since late 2023. And certainly this epizootic in wild animals has been happening since then. But the humans, why not? Sure. Or cows.
Cows.
And we have a report on measles from the CDC. We now have 128 measles cases in 20 jurisdictions. So that's the new word for state? Because it includes Puerto Rico,
because
right?
It could be Puerto Rico, Guam, Wake, etc.
Yeah, well, those things, okay.
Yeah, so they refer to jurisdictions. In this case, it's all states. So
70 of these people have been hospitalized so far in this outbreak. So this is not trivial. So it's increasing. And also in England, we have an article from the UK. Measles is at its highest in the UK in a decade. Now, you may say, why is this? Well, when people are reluctant to vaccinate, which seems to be a trend these days, wouldn't you say that? Isn't it kind of a trend in certain groups? Measles virus is one of the first to be seen because it's highly contagious, and one person can infect many others. It spreads in very small droplets of respiratory excretions. So this is why we're seeing them first. But it's other viruses will come back as well. According to Paul Offit, this is a pushback from the mandatory COVID vaccination. Now parents are saying, I don't want my kid vaccinated. So, almost every state has a religious vaccine exemption that you can choose to take, and the parents are saying, no, my religion says I can't take a vaccine. And, the validity of that is questionable. But Paul said there's only one religion where it's actually written in the whatever the document is for the religion that you can't vaccinate yourself. The others is just using it as an excuse, in my opinion. Anyway, that's bad. You know what? Measles can be bad. So I don't understand why you wouldn't immunize your kid. What do you think measles vaccine is going to do? It doesn't cause autism. It's been shown for decades. Come on, folks.
This isn't about facts.
Oh, right.
No,
This,
not at all. Vincent, I want to go back to something you said about the H5N1 in cows. The only thing I saw a good excerpt or executive summary of that on PBS last night, and
Yeah.
all of the people that were giving information were clear to say that all they found were sequences of the virus. They did not find an entire viral particle. They did not consider. They tried to use milk as an infectious agent
Right.
to birds, and it didn't work, of course. And they
Yeah.
said these are, and then they couldn't explain where it came from, of course. So, if you
Well, the
think about
cows are infected. No, the
farms,
cows are infected. They make
yeah, but where did the cows get it though?
the chickens.
yeah
And where did
birds,
the chickens get it?
birds,
Deer.
ducks.
Birds. Yeah, I mean, the chickens, we knew it was in chickens.
Yeah. Okay.
I mean,
Or
wild
migrating
birds,
wild
Or
birds.
migrating birds who happened past the
Flyovers.
dairy herd. Yeah.
So the farms in some areas are collection areas for migrating birds, and it's true particularly in Israel, where you have a lot of farming in the surrounding, and it's all desert. And you've got these wild birds coming out of Africa, and that's where the West Nile virus first started, I think. But think of all the food that's available on a farm to the animals, and there are other animals out there that want to take advantage of that as well. So that it's very easy to make the association as to where it might have come from.
Yeah, the milk doesn't have infectious
No,
virus in
not
it,
at
but I'm not worried about the milk. I'm worried about the cows infecting
No, but
people.
people might get the
Right.
wrong idea
But in
because they didn't hear
Ohio,
the whole truth.
I was just in Ohio, and they have a lot of infected cows there, and then people are getting infected, but they said they're mainly getting conjunctivitis.
And they're people who work with cattle.
Yeah, they work with cattle, right?
Right.
Like you guys work with me, and I work with you, and there are people who work with cows. I wonder
But they
what
do.
it would be
They
like
don't do
to
it
have-
over a video conference.
Where seldom is heard.
We could get we could work with cows here at Microbe TV, but we don't have a lot of room.
We
No,
don't have.
they
We could
wouldn't
use mini
fit.
cows though. They have little mini cows.
They have mini cows, really?
They do.
What do you do with a mini cow?
Tiny little cows. They have mini pigs, mini cows. They have
Are they pets?
You get tiny amounts of milk.
you get that's right. You get half pints.
By the way, I wanted to in the CDC page about measles. There's a chart on vaccine coverage. So there's a map of the U.S. and it colors each state according to vaccine coverage. And so measles requires 90 to 95% or more coverage with vaccine to prevent outbreaks. And so California, Montana, Nebraska, Maine, New York, a bunch of states around here.
Massachusetts.
Connecticut, Rhode Island, Virginia, West Virginia, Delaware, Maryland, Tennessee, and Alabama. Are you guys proud that I could name them by looking at
Very
the
good.
shape?
That's good identification of the jurisdictions.
The jurisdiction. So they're all 95% plus, but then we have a lot of states that are 90 to 94. And then we have a good number, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 states that are less than 90%. And that's a recipe for measles outbreaks. Come on, states like, what would that be? Georgia? Ohio, for sure.
Wisconsin, Iowa, Minnesota. Yeah,
Idaho.
Georgia, Oklahoma, Colorado, Arizona, Idaho,
Alaska,
and
and Hawaii.
And Hawaii. Oh, yeah.
So, if anything, this proves that Alan and I can recognize states by their shape.
There's also, I cheated a little bit with Iowa because I knew it was one of those rectangular Midwestern states. And
you hover over it.
And I hovered over it, and you get
I
the
didn't
state
do
name.
that. I didn't do that. But anyway, check that out. That's really good. All right, we have a snippet and a paper for you today. And the snippet is a Nature Communications paper called Positive Selection Underlies Repeated Knockout of ORF8 in SARS-CoV-2 Evolution. And the authors are Wagner, Kistler, Perchetti, Baker, Frisbee, Torres, Aragona, Yoon, Figgins, Greninger, Cox, Oltine, Roy-Choudhury, and Bedford. Trevor Bedford is the senior author, and we had Trevor on TWiV last summer at ASV.
Vincent, I think you said ORF5. I think it's ORF8.
Did I say ORF5?
You did.
I wonder why I would do that.
I don't know.
Well, it's ORF8.
It is ORF8.
It's ORF8.
Now,
And this and the other paper we'll be talking about are both open access.
And the ORF stands for Open Reading Frames.
So that's the first thing I wanted to mention. This is a protein encoded by the genome of SARS-CoV-2. And now proteins can have cool names like spike and nucleocapsid protein because we know what they do.
Right.
But ORF8, we didn't know what it did, so we just called it an open reading frame.
It's an open reading
8.
frame in the sense that it could be translated into a protein. We now know that it is.
Exactly.
And so
It
we
is
just
made
called
into
the
a protein.
protein.
There's a
ORF8.
start codon, there's a stop codon, all that stuff.
Right. Now, I've been interested in ORF8 for a long time because I heard a talk by Christian Drosten back in 2019 or 18. I don't remember when. It was in Rotterdam at a virus meeting. Well, he talked about the fact that this ORF8, early in the SARS epidemic in 2002-3, right? ORF8 was lost, and all subsequent isolates had a deletion. And there was some suggestion that maybe it was less virulent in people also. But, it was lost early on in the outbreak. SARS-1 didn't go very far. It went 8,000 cases. But, and I blogged about that in January of 2020. The COVID was just getting going, and I remembered this, and I heard that SARS-CoV-2 had an ORF8. So I said, it'd be interesting to see if this thing gets deleted in SARS-CoV-2. And that's what this paper is
completely
about.
different ORF8 than the SARS-1, though, I presume.
Well, it's a similar protein. It's an accessory
A similar
protein.
Okay.
Yeah.
Similar protein.
So this paper is all about, it turns out that now, I think over 90% of SARS-CoV-2 isolates have lost ORF8.
Yep.
So this is a computational paper which tries to understand what is driving the loss of ORF8.
Right. So they present at the beginning 3 hypotheses. This could just be that ORF8 happened to be deleted in the context of some other mutation or whatever that was advantageous, and so it hitchhiked. It could be that it was like a neutral change.
Yep.
Or it could be so it could be that it's linked, hitchhiked. Basically, the first two hypotheses are very similar. It's different scenarios where it would hitchhike. And the third is that deleting ORF8 is actually good for viral fitness in humans.
Right. Yeah, so the deletion would be improving the fitness. And so this paper looks at that. And if you're wondering, ORF8 protein has been studied a lot. And it's not just in, this deletion is not just in SARS-CoV-2, but also in pangolin coronaviruses and mink isolates.
Oh, okay.
So it's basically an immune evasion protein. It's got a lot of functions, like it helps downregulate major histocompatibility class 1 molecules from the surface, which would be an immune antagonist, and it decreases a lot of other immune activities, and they think it might even contribute to cytokine storms by activating what we call the IL-7 pathway. So it's involved in immune evasion. Okay, so it's not essential for virus reproduction. You can take it out and the virus will still reproduce, although in a host it may have an altered interaction with the host.
But given all of its effects or apparent effects on immune evasion, it's not the kind of gene you would expect to go away. Seems like it ought to be useful.
For sure, an immune evasive gene would be very useful for a virus, yeah. But unless that usefulness is balanced by some negative fitness impact or something.
Right, like
that.
maybe that's useful in, what they're going to get to later in the paper is maybe that's useful within the host, but it inhibits your ability to spread between hosts, in which case
there'd
be a pressure against it.
So that's that's very interesting because T-cell escape mutants of viruses are only useful within hosts because the way that works is that you involve MHC molecules, and that's so polymorphic that a CTL escape mutant in one person is of no use to someone else. So it's only good in host. But that's fine because those are long-term infections of many years. So for T-cell reactivity to be lost can aid the virus. But so that's a real-world example of that. So what they do here, it's a computational analysis of sequences of SARS-CoV-2 from Washington. I don't have to say state. It's Washington. If it's D.C., we say Washington, D.C. If it's Washington, it is the state known as Washington. Why do people have to write Washington State, Alan?
Because they're from D.C. and they think Washington means D.C. And we should point out all of the authors are from Washington.
Yes,
Full stop, just Washington.
Trevor Bedford.
University of Washington, Fred Hutch, and HHMI.
They did a lot of sequencing.
Seattle.
Remember, they had the first U.S. case diagnosed,
yes,
right?
And in the Washington Department of Health.
They did a lot of sequencing, so they have great coverage. So this paper, they take sequences from February 2020 to March 2023. And it's emitting Omicron because Omicron had its own incredibly powerful selection. So they look at all the others. So the idea here is to understand what is, as Alan said before, what's driving the loss of ORF8. And so they have great sequencing coverage here. And so they look through all they have 14,929 samples with a potential knockout of ORF8. And there's a lot of trickiness about identifying knockouts, which I won't go into, but it's not so straightforward as you might guess because there's sequencing errors and so forth, and you just don't know. Most of the knockouts were found in variants descending from alpha. Remember alpha, the very first variant to arise? Those variants have most of the loss of ORF8. And loss of ORF8 occurs by the introduction of a stop codon, a premature stop codon, right? And this is, ORF8 is made from its own mRNA, so it doesn't matter if
it doesn't affect anything downstream.
There's no downstream.
So, Vincent, this happened sometime after the pandemic, of course. So do they pinpoint exactly when it lost the ORF8?
Well, some lineages lost it and some didn't.
Oh.
So it's not universal. It's there are lineages where it lost it and lineages where it didn't. And what they do subsequently, which I think Vincent was probably about to get to, is they look at those lineages and how successful they were subsequently, right?
I see.
Yeah, I think it was quite early in the pandemic when you started to see the loss. I don't remember. Let's see if this tree does.
Yeah, it hasn't been fixed in the viruses spreading in humans. So what was the figure? 90% of circulating SARS-CoV-2 has ORF8 knocked out, but that means about 10% doesn't.
Yeah, some of them still retain it, yeah.
Hmm.
All right, so they look at these thousands of samples, and I said most of the derivatives of alpha have the loss. And most of these losses are by premature stop codons. So basically, and most of the stop codons occur very near the end terminus of the protein. I think about 26 amino acids are left before the stop codon. So there's no big piece of protein left that could have some function. It's 26 amino acids is not enough to do anything. They compare the loss of ORF8 to other genes in SARS-CoV-2, and this is knocked out way more frequently than any other gene. Some other genes are lost, but this one is way more frequent. All right, so if a gene loss is deleterious to the virus, it should be eliminated, right? The loss shouldn't occur. It shouldn't perpetuate through the virus population. And here, as they say, they look within the host, because if the high rate of knockout in the sequences they're looking at, remember, these are consensus sequences from people, is also seen in host, within host. This would argue against deleterious fitness associated with the knockout. So they have some sequences from single hosts where they can look at the knockout, and they find that in fact ORF8 is also lost within host as well as outside of the host. So that takes care of that hypothesis. The other thing they do now for most of the rest of the paper is to look at positive selection. So is the knockout good for fitness? So is it selected for? And
so is it good or is it just not bad?
Yeah, is it good or is it just not bad? Right. So they use
anthropomorphic.
They use a computational method which looks at synonymous versus non-synonymous changes, right? So these are amino acid base changes. So a base change that doesn't change the amino acid is a synonymous change. You could think of it as a synonym, right? And a non-synonymous change changes the amino acid. So they look at the ratio of non-synonymous to synonymous changes, which you can just do by scanning the sequences with a computer program. And so, for example, if the dN over ds is more than 1, that's indicative of positive selection. So that change is maintained in all the different isolates that you have. If it's less than 1, it's negative selection. So it's not good, and it's being selected about. And 1 is neutral evolution. So if the top and bottom are equivalent, it's neutral evolution. So they look at the ORF8 and ask what's going on there. And ORF8 has a high value, greater than 1. And it's higher than that of any other gene, which suggests positive selection. And then they say, well, there's some caveats here, so let's address these caveats. And even after addressing the caveats, it still looks like the ORF8 deletion is being positively selected. So in other words, it's of some benefit to SARS-CoV-2. What that might be, we don't know, but that's what the genome is telling us. They then quantify that. They look at lineages of virus that have the ORF8 deletion, and they find that these are larger than ORF8 with a than clades with a synonymous mutation in ORF8. So they're the ones with the with the deletion are larger. Plus, they circulate longer. 11 and a half days for the ORF8's deletion versus 7 days for a synonymous mutation in ORF8. So we're comparing a deletion versus a synonymous mutation, which is a little different, but they said we can do this. We know what we're doing. Don't try this at home, but
they have a biostatistics license. They're allowed.
They also look at the rate at which these clades with ORF8 deletions grow. In other words, you have an initial one in an area, and then how fast does it propagate? And the clusters with nonsense mutations in ORF8, that is, that lead to deletions, grow 6.3 times faster than those with synonymous mutations. So the stop codon is a non-synonymous mutation, obviously, right? Because it changes the amino acid to a stop codon. And that's why they're treating it as a non-synonymous mutation. It's totally fine, yeah. And so they say this tells us that the knockout is somehow boosting virus fitness.
Yep.
Okay? So some function of ORF8 is not good for fitness. Let's see here.
They do then also discuss the possibility, they can't really sort this out, but so ORF8 may have an advantage within the host and a disadvantage spreading between hosts. That's the hypothesis, and they present some previous work from other groups that supports that notion.
Yeah.
And then the idea is that in a chronic infection, which is, we think, maybe the source of a lot of new variants, you keep ORF8, the virus keeps ORF8 because it's advantageous in the host, and in the chronic infectious state, the virus is more pressured to continue surviving in that host than to spread to the next one. And that may be the reservoir that prevents the ORF8 deletion from getting fixed in the population.
Right.
So that may be the source of new ORF8 coming in.
They also note that many changes in spike are positively selected for, and they're associated with fitness gains. In fact, the rate of growth of those compared to ORF8 are similar. So they say this is more evidence that maybe the ORF8 is positively selected, right? And they also look at whether it's found in one clade versus another, right? One variant or one clade versus another. And there's some clades that mostly have it, but it's also found in others as well. So there's no clade-specific association of that.
And the deletion may decrease pathogenesis. That's, again,
right.
They cite some various evidence on that.
Yeah, so people have suggested that it reduces clinical severity. So then they say, all right, let's take our genome sequences and link them with outcomes in the disease reporting system. And they do this again with pre-Omicron because the Omicron itself was thought to be
different story.
Story. So 8% of individuals infected with intact ORF8 were hospitalized. 6.7% with ORF8 knocked out were hospitalized. 1.8% of individuals infected by intact ORF8 containing virus died, compared to 1.3%. These are very small differences. Yes. But I want to remind people, they do not take into account population immunity, age, comorbidities, which could all influence, this. So I think this needs more study, frankly. I mean,
it may be too late to study that.
It may be too late. In fact, yes.
I gather that no geographic distributions were noted, or age, or
well,
sex,
this is all a study from Washington, right?
Well, no, they did look at international samples, and I don't think they found that there was any striking, like if you're in a particular place, you don't get ORF8 deleted. It just seems to be consistent.
Yeah, but most of their data is from Washington State. But I think that's correct, that it's found elsewhere as well.
I had the unfortunate event happen to me at the ASTMNH meeting in Seattle.
Yes.
I caught, and so did you, Vincent. And we
both
did. Acquired. I wondered if we had an ORF8 deletion or not.
Good question. Maybe. What was that? December 2022?
As we know exactly when it was, because the woman in charge of the exhibits came around and shook everybody's hand the morning of the opening events. The next day she phoned up and said, "Oh, by the way, I just tested positive for SARS-CoV-2. Have a nice day."
All right. So all of this, Dixon, have you followed this so far? You're kind of the lay person here. You got it?
I got whatever there is to get. I mean, I don't understand why the deletion occurred. I don't understand why the deletion is still there. And I don't understand why in the same populations there is no deletion.
Well, it's not, first of all, it's not a deletion of the gene. It's an introduction of a stop codon. So the protein is gone.
Well, that's what I meant.
It's not really a deletion in the genome, right? It's a knockout of the protein.
That's fine. That's fine.
So it's best to say ORF8 null virus. It arose just because random changes happen all the time.
Well, why doesn't that happen to the whole damn virus and just get rid of it then?
Well,
that's
right,
not
because the selective pressure.
No, I understand
that.
You're trying to find an argument why this happened.
Right, that is something that remains a little up in the air. So all this paper shows, which is important, is that there seems to be a positive selection to eliminate the ORF8 protein in human infections.
Exactly.
For SARS-CoV-2. And so there's some selective pressure that's causing the virus to lose this protein repeatedly. And that's worth investigating further.
Yep.
Oh yeah.
Now, they do speculate a little about what the mechanism might be.
Right.
And I think this is interesting. So they say that ORF8 protein interacts with spike in the endoplasmic reticulum, okay? And it reduces the display of spike on the virus particle's surface. So, and they've shown that in experiments that it leads to less spike on the surface of the particle. And what they say, that could give you better fitness by avoiding the host immune response, because if you have fewer spikes on the particle, you're going to be less able to be neutralized by antibodies, right?
But you may be less able to transmit to the next host.
Right.
Because you have less spike on the viral particle, and when you go through a bottleneck of transmitting from host to host, that may affect the ability to establish a new infection.
Now, they did find an elevated within-host rate loss of ORF8 compared to outside of the host,
Right.
but they say, you know, we these are all acute infections, and we may not be seeing the effect of loss there. Then they say, you know, these knockouts have not been fixed. First of all, they've not been fixed, only 90%. And they've only spread in alpha and XBB subclades, and they don't know why that is. But one thing they do say is very interesting. They say this could knock out the loss of, and I don't think we should say knockout because it implies,
this
I guess
protein
it's a
loss.
This protein loss is an example of what they call clonal interference. Okay, so an example of that would be that, so here's how it would work. XBB descendants have ORF8 knocked out. Then suddenly BA286 outcompetes XBB, and ORF8 doesn't matter. It's not going to help as much because BA2.86 is so much more fit than XBB that having ORF8 knocked out doesn't matter. And so they say that could be why ORF8 is not present in all lineages. And they also say, I think, Alan, you mentioned this, that these ORF8 changes are not present in chronic infection-associated infections, right? And they say maybe because having intact data allows persistence and evasion of the immune response, because you have a long-term infection where immunity is going to make
different pressures.
Different pressures, yeah. So they say they hypothesize disproportionate contribution of chronic infections to global evolution could prevent ORF8 knockout fixation. So there's a good fraction of infections that are chronic and immunosuppressed people, and so that could be the reason. But you know, these are immunosuppressed people, so I'm not sure how much of an immune response you need to evade, right?
There's probably some, and it depends on the patient, and yeah,
they're all different. That's true. You just have to be careful when you speak in these global terms that you are really, there's not a complete absence of an immune response in every immunocompromised patient. It's different kinds, and so that may be where ORF8 is doing it. So I think this is interesting and tells you that there's probably positive selection for ORF8 loss. And now what do we do? Well, there's not much we can do. This is an interesting problem, and it's a virological problem in my view. I don't think it helps in any way of surveillance and epidemiology. You just get vaccinated and you'll be protected, right?
Yeah. The longer term way I see this helping, I mean, of course, it's always best to know more about a serious disease causing virus. But I might imagine if we find out that ORF8 is involved in the cytokine storm, we already know that is one of the deadliest parts of this and many other viral infections, and maybe we could target that and develop something that's going to stop that aspect of it.
Yeah. Dixon, you have any other questions?
No, and I don't have any answers either. I think the best thing to say at this point is stay tuned.
Yes.
Dixon, what's the most successful parasite of humans?
Toxoplasma.
I thought so.
Yeah. In fact, it's the most successful parasite, period. It infects all mammals.
Someone had another answer, and I said, no, I think Dixon would say toxo.
I would definitely say toxo.
Okay. I forgot which one they said, but...
Pinworm, maybe. I don't know.
No, it wasn't.
It's not a parasite.
I forgot what it was. All right. So I learned that from you anyway, that Toxo is very
Well,
successful,
I'm glad I could contribute.
among other things. Our next paper is also in Nature Communications. How
It
about that?
is.
How about that?
Now, since we've been talking about influenza H5N1 viruses, I thought we would talk about some other influenza virus. And this is cross-species spillover potential of the H9N2 bat influenza A virus. Very cool. H9N2.
And bad influenza virus.
Bat influenza. Did you know that bats could have influenza viruses? Well, now you're going to learn. The first author is Rabeh El-Shejni. I don't know how many co-first there
It's
are.
only one first author and only one senior author, Richard Webby.
Richard Webby.
I do want to call out the third from last author, Muhammad Ali. I'm sure is not the same person.
I think Muhammad is spelled differently, but
Yes, it is.
here.
And this group is from a whole bunch of different places, all over the world. A National Research Center in Giza, Egypt. So there's someone from Giza and someone from Memphis, but that's the Giza in Egypt and someone from Memphis
Exactly.
in Tennessee, St. Jude Children's Research Hospital. Someplace
Cool.
called Human Link in Dubai, and I don't know if that's a company or a research center. High Point University in High Point, North Carolina, University of Hong Kong, and Scripps in La Jolla. So really
Dixon,
very global.
have you been to Dubai?
Yes,
Is it
I
good?
have. Well, I wish I had been there during the flood. That would have been an interesting time to experience an all desert phenomenon that has never happened before.
Yeah.
But yeah, I was there when the Burj Khalifa building became the tallest building. I was there for the Tall Building Council
Oh,
meeting,
cool.
and it was quite an amazing building to look at. It looked like from a distance, it didn't look like a building. It almost looked like a fountain pen. It was so skinny, and so they must have done oodles of wind tunnel work just to make sure that the damn thing wouldn't fall over. And I was impressed with that, and I was also impressed with the fact that there were a lot of buildings in Dubai that had their family names on them, and the buildings did absolutely nothing but just show off the fact that they had a lot of money. That's all. So, unfortunately,
Right.
that's still the case.
So the senior author, Richard Webby, will be on TWiV at some point. I think we're finding a date for him to talk about H5N1.
Great.
So that'll be
Cool.
fun. All right. So most influenza viruses, they're in people, and the reservoir is waterfowl, right? The major reservoir are waterfowl. Many mammalian hosts are infected besides humans, dogs, horses, cows, pigs, etc., cats. But they've been recently found in bats, and we covered this first paper on TWiV a long time ago, 2009-10. The first influenza virus of bats was found in a little yellow-shouldered bat in Guatemala. I just love the name, little
Yes.
yellow-shouldered.
Many bats have really charming names.
Totally different from all other known influenza viruses. Even the HA and the NA were very different. And then another one was found. So that one was called H17N10. And in 2010, another one was found in Peru in New World flat-faced fruit bats. And this was called A18N11. And both of these bats were asymptomatic. They weren't sick, and they got these influenza viruses from them. Now, the cool thing about the hemagglutinin of
And the H18N11 is also not related to any other,
right?
These are like totally bizarre, came from Mars flu viruses.
And that paper we did on TWiV. Now, as I mentioned earlier, the receptor for the hemagglutinin of these viruses is not sialic acid, as it is for most other influenza virus that we know of, but it is a major histocompatibility molecule, class 2, and that was discovered by a group including Silke Schurk, as I told you a while ago. She was going to factor in, and she's going to be at one of those meetings. So they use a proteinaceous receptor rather than a sugar to enter cells. And these viruses in the bats were detected in rectal swabs. So maybe a fecal-oral transmission rather than respiratory. Got that, Dixon?
Yeah. So up to
Yes.
that,
I just
so
want to make sure.
up to that point, it was pretty much bat influenza viruses are off in their own department. They use a different receptor. They've got these distant relation
There you are.
H and N, and then 2017, the virus in this paper showed up.
Now, this viruses, H17 and N10 and H18 and 11, bats are highly seroprevalent in Central and South America, but not in Europe. So this virus could be restricted. Now, the virus of this paper was discovered in 2017 in Egypt from Egyptian fruit bats, and it's different from H17N10 and H18N11, but it is within the H9N2 subtype, which is known to be avian influenza viruses. So the HA and the NA are distinct, but the HA and the NA are H9N2, right? That's what you get that from. And the remaining genes come also from avian influenza viruses, it looks like. It was first found in oral swabs of the bats. They could be grown in chicken eggs, unlike those bat viruses. And inoculating fruit bats with the virus in the lab gives you an infection. So, and also some bat-to-bat transmission seem to have occurred in that. So you got now bats have two distinct forms of influenza viruses. You have the H17, H18, which are very distinct genetically, and they bind a different receptor. And then these and 9-2-like viruses, which they say the simplest explanation is that it came from a bird into a bat, and now it's established in a bat. So if I were a bat, I would start working on vaccines. Well,
and stay away from the birds.
Stay away from birds. Dixon, do birds and bats ever mix?
They must, because the flyway from all of Central and Western Africa goes up through the Nile River. So and there's no water anywhere else.
Right.
So, and well, the Lake Nasser, of course, but yeah, the birds are. I was surprised to learn that they had fruit bats in Egypt. I would have thought every piece of fruit is so valuable that people would pick it before the bats got to it.
Apparently, there are remote
Not the
regions
case.
where the fruit
Dates,
bats.
maybe they eat dates. Do they have any
I don't know.
feeding preferences?
Don't know. All right, so this paper is going to look at this bat H9N2 virus and try and know if it's a threat to people, as far as we can know. Right?
Yeah, because this is a familiar looking flu virus, and it appears to have come from birds into a mammal, and that's kind of the pattern you look for a new pandemic flu virus.
And this one binds alpha-2, 3 sialic acids, which is what avian influenza viruses typically do, right? So they're going to do a number of experiments. Now, the first one, they look at the pH inactivation of the virus, and basically incubating viruses at a range of pHs. And what they find is that this Egyptian fruit bat virus, which we'll call H9N2, has resistance to inactivation at a pH similar to that of human influenza viruses. So pH resistance of influenza viruses tracks with pathogenicity and transmissibility, and the resistance pattern of this virus to pH is similar to that of human viruses. So that's kind of interesting. They look at the neuraminidase of this virus. Now, the neuraminidase is an enzyme on the surface of the virus particle whose function in most influenza viruses is to cut sialic acid from proteins to allow the virus to spread after it's released from cells. And the neuraminidase of H17N10 and H18N11 does not cut sialic acid. It doesn't have what we call sialidase, cutting off sialic acid activity. But this one does. This H9N2 from Egyptian fruit bats does have sialidase activity. So, and that's makes sense because if you bind a sialic acid, you have to have a neuraminidase to cut it off. Otherwise, the virus that's made will all stick to cells once it's produced, right?
And the other bat flu viruses probably don't need that because they're not using that receptor.
No, they're using a proteinaceous receptor. It's right. Why viruses like that don't need something to cut off their receptors is unknown. But no, poliovirus doesn't have anything to cut it off of the poliovirus receptor. I guess it's not an issue, right?
Yeah.
Must not be an issue. All right. So we know the sequence of the hemagglutinin of this virus. And they said previously people showed that this hemagglutinin has the receptor binding residues typical of those HAs that bind alpha 2,3 linked sialic acids. And they just do more studies on binding to different sugars and confirm that this receptor specificity of this H9 hemagglutinin is similar to avian viruses that are found in North America.
All right. So the alpha 2,3 linked sialic acids are common to birds, and the alpha 2,6 linked sialic acids are more common to humans. So normally you see a shift from alpha 2,3 to alpha 2,6 in going from birds to mammals, but these viruses don't seem to have had to make that shift. They're still alpha 2,3 avian-style binding, but they're obviously infecting mammals.
Right. All right, so now we're going to do some infection experiments.
And by the way, I love that this is a fully modern, interesting, important paper that's like mostly classical virology experiments.
It is.
I mean, you could have done these experiments 30 years ago if you'd known to look for this virus. It's just great.
It's very good. I think this underscores that we still need to do certain kinds of experiments.
Yeah.
So they want to know how these viruses reproduce in human cell culture. So they make air-liquid interface cultures of human bronchial and lung cells. And so what that is you take bronchial or human lungs or lung cells from people, you plate them on a membrane, and then you take the membrane is suspended in the cell culture dish. So there's medium underneath it, and then the top is exposed to air. And then the cells like being in air because that's what they do in
They're
you.
lung cells.
They're lung cells. So they differentiate into all the right cell types. It's very cool. That's why it's called air liquid, because the liquid underneath gives them nutrition, and then the air, they're like, oh, finally something to breathe. And then they infect them with the H9N2, and they also infect them with an H5N1 and a duck H9N2 as representatives, and then a human 2009 H1N1 pandemic virus as a representative human virus, right? So the human virus replicates really well in these human cells, as you might expect, but all the other viruses of bat origin replicate to lower cells, as well as the ones from avian sources. So this is what you would expect for a human versus a bat or a bird virus. So this is called ex vivo replication efficiency because you're doing it in cells and culture. So that's a good word. We are using this in the textbook because, there's in vivo, in vitro, and then there's ex vivo and ex vitro. I don't know if there's ex vitro, but ex vivo is not in an animal, but it's in cells and culture.
In cells, it's in primary cells in culture, right?
Yeah.
So these are not passage cell lines. There's ex vivo just taken out of
the host. That's right. So you may wonder how you get bronchial and lung cells from patients. Well, people have bits of their lung taken out all the time in their bronchi, so those are available. Yeah. Okay. Next, they look at alveolar epithelial cells. So the alveoli are the cells lining the alveoli. Duh. They're all the way down in the lung where the air exchange occurs from the air into the blood. And so these are called primary human alveolar epithelial cells. And the avian virus, they say there's not much difference in titers among all of these except for the H5N1. So they're all replicating similarly in those cells. And that's interesting because that's where humans have most of their alpha 2,3 linked sialic acids, way down in the bronchi and the alveoli, right? So maybe that's part of the reason for that. So then they look at what cytokines are induced by these viruses when they infect cells. And why is that interesting? Well, many of these highly pathogenic viruses induce a cytokine storm in people, and that's part of the severe pathogenesis. So they want to say, can we learn anything from this? And they say the H5N1 induced the highest levels of cytokine mRNAs, cytokine and chemokines, and the bat Egypt had the least mRNA encoding chemokines and cytokines. So the H5N1s, when they infect people, they cause cytokine storms. That's the basis of severe disease in many individuals. So this is saying that bat virus isn't there yet, right?
Which is good news.
That's good news. Now, how about pathogenicity? We're going to look at two different hosts for this virus, mice and ferrets. And remember, mice lie, monkeys exaggerate, and ferrets are not humans.
Right.
Which just tells you that mouse and animal models, you got to be careful about them. So they inoculate mice with the bat virus and also an avian H9N2. So it's another H9N2, but it's from birds. And the bat virus was more virulent than the duck virus, caused more weight loss and more virus reproduction in the lungs. And they confirmed that by immunostaining lung sections with antibody to the viral nucleoprotein, and you can see diffuse replication throughout the lung. So that's interesting that the bat virus is more...
and at higher titers, the bat virus kills the mice, whereas the duck virus does not.
That's right. They met human endpoints at doses of 10,000 and above, EID 50. All right, but we don't know what that means because these are mice.
These are mice, yes.
But that's what we have.
But it's a mammalian model that is straightforward to use, and you took this virus from bats and you put it in mice, and the mice die at reasonable inocula of this virus, and that's something to look out for.
But the ferret model, it gives you, according to WHO, that what happens in ferrets is important for pandemic risk assessment, right? So they do some ferret transmission studies. So they put, they'll infect a ferret and put it in the same cage as another ferret and see if the virus is transmitted. So that would be contact transmission because, ferrets can't resist touching each other. And then they put ferrets in a separate cage, uninfected ferrets in a separate cage. So you would need to have aerosol or respiratory droplet-driven transmission and see if the recipient gets it. Then that would be a more reasonable model for human transmission.
Well, it'd be a more reasonable model for human transmission in public, whereas the direct contact is more like a household.
Humans contact each other?
So I've heard.
Okay. Yeah, I don't even shake hands anymore.
No, right.
So they infect ferrets with either the bat H9N2 or the duck H9N2. They measure the virus in the nasal washes. They say both viruses called mild clinical symptoms in signs in infected animals.
Right.
There's a sign. A symptom is
very, very mild. Like the weight loss is not noticeable at all. They see maybe a slight decrease in body temperature, and I mean these ferrets are fine.
Ferrets have symptoms, but they can't tell you about them, so we don't know what they would be. So we measure signs in them. The bat H9N2 transmitted to contact ferrets by 2 days, but the duck H9N2 did not.
Right.
So and the bat one transmitted both with direct contact and airborne contact.
Very interesting.
Yes.
Right? So that, is a little bit concerning because as they say later, we'll get to that in the discussion. The WHO looks at ferret transmission as an indicator for pandemic threat. They took these animals apart after the experiment and looked for virus in different tissues. They could find it in the lung and the nasal turbinate initially at 3 days, and then at 5 days they find trachea, brain in addition. And they also did immunostaining to confirm these findings. They also looked at, this is a new one, replication and transmission in mallard ducks.
Yes.
So they infect birds with both viruses, the bat H9 and the mallard duck H9. No clinical symptoms for 2 weeks. The bat virus did not replicate in the ducks. They got no virus out, no seroconversion, but the duck virus did replicate. It was found in cloacal swabs. That's the other end of the bird.
Yes.
Right? In the bat, it's a rectal swab. So in a duck, it's a cloacal swab, right?
Because ducks have one hole for everything.
There you go.
Is that right?
Yeah, so that's for peeing, pooping, and reproduction. It's all through the cloaca.
So would that be like one hole to rule them all?
Yes.
Indeed.
Dixon, you got that cultural reference, didn't you? Pop cultural reference.
Yeah.
All right, so
I'm not that old.
it's interesting. So the duck virus replicates in ducks. The bat virus does not. And that's the story here.
Right.
So let's just chat a little bit more. The idea, again, that this virus most likely came to a bat from birds, perhaps ducks. But H9N2 in general, they say the H9N2 viruses became established in land-based poultry in the 1990s. And many isolates have been taken, which they, and these viruses taken from land-based poultry, these H9N2s, have an amino acid change in the hemagglutinin, which allows the hemagglutinin to bind alpha-2,6 sialic acid. So it's a signature for human receptor specificity. Remember, the avian hemagglutinins bind alpha-2,3 sialic acids, the humans alpha-2,6. So these viruses from chickens, these H9N2 viruses from chickens, have human receptor binding specificity.
Are there any non-land-based poultry?
Poultry?
Because poultry, I think, is domesticated birds, right?
I don't know. What is poultry? What is poultry? So many good questions. Poultry is domesticated birds kept by humans. Yes.
Bingo.
So they're all land-based.
They're all
right?
land-based, yes.
Yeah.
Okay.
Well, a duck is a waterfowl.
It is.
And in China, they raise them in ponds.
Yeah.
By the way, I think to assess whether or not the mallard ducks actually were suffering or not from the infection, they should have sent them to a quack doctor.
Yes, of course.
Quack
No,
duck.
they should quack them open and look.
They should quack them out.
That's
That's
good.
right.
I like that.
Exactly.
I like that very much. All right. So the bad viruses that the bad H9N2 that we've been talking about in this paper has a glutamine at amino acid 226 of HA, which is a signature of alpha-2,3 sialic acid binding. The human alpha-2,6 binding HAs have a leucine at 226, so you can sequence it and know immediately what the binding is going to be.
Mhm.
And that, consistent with that, the bat H9N2 binds mainly to alpha-2,3 linked sugars.
But that's one amino acid change away from being able to bind to the other.
Yeah. Yeah,
Exactly.
so it's one amino acid change. I don't understand why the chicken H9N2s have all the human signature in them. I'm not quite sure. Maybe we'll ask Robert Webby that.
Yeah.
Is that his name, Robert or Richard?
Richard.
Richard. Thank you very much, Dixon. Thank you, Richard Webby. Okay, so they say little brown bats, Myotis lucifugus, are widely throughout North America. They have both alpha-2,3 and alpha-2,6 sialic acid throughout their respiratory tract. So this makes sense that this virus can reproduce in bats, right? And they say there's no selection for alpha-2,6 because bats have both. So if you already got a virus with alpha-2,3, it's not going to change to alpha-2,6 unless there's a fitness advantage, and apparently there is not. So these viruses, these H9N2 bat viruses, have avian like HA receptor signatures, and they say the replication in primary human cells are also avian-like. They don't they're low replication compared to the human viruses,
Right.
which replicate to higher titers. But in the when you go down into the alveoli, there's little difference between the replication. Where there's alpha 2-3, mostly sialic acid. So that's interesting. All right. So what else here? This says here the robust transmission of this virus to animals, ferrets, does imply an elevated risk to human health. The ability to transmit between ferrets is a highly weighted element in the WHO's tool for influenza pandemic risk assessment. So that's part of the...
And the influenza risk assessment tool that the CDC uses, which I assume is very similar.
It has different acronyms.
Yes, so you can use the TIPRA or the IRAT, but...
oh my gosh, so many acronyms.
Oh, if you interact with any government agency, it just gets completely out of control, yeah.
That's true.
Why do they like acronyms?
Because they've got all these programs that have names and they have to
They go
abbreviate
on.
them
I see.
because they...
That's
Why
right.
do we have jargon? I mean, why do we refer to RNA and DNA instead
Yeah.
of spelling,
right.
giving the whole name? It's
It
'cause
saves time.
It saves time. We speak in a shorthand, and
But it
people in the policy
excludes
world
a
do
lot
the
of
same
people.
thing. Yes, but then if somebody needs to communicate outside of that sphere, it becomes very alienating.
So
You know, like here, you mentioned a term before, Vincent. You use CTL.
yes.
Cytotoxic lymphocytes.
Lymphocytes, right.
If you just, but it takes longer.
Yep.
That's all.
Yeah, but I should have said cytotoxic T lymphocytes
Oh,
Because we are trying to explain to the public, yes.
Yeah.
Don't worry about it.
All right, so
People
the
outside of
conclusion,
virology.
the
I say it that
conclusion,
way all the time.
Yep.
the conclusion here is that the phenotypes of this bat H9N2 virus has some phenotypes of avian viruses, some with human. They say this mix makes it difficult to assess the pandemic risk of this virus. They say we should, they say, further investigating the abundance and nature of influenza viruses in bats in comparison with other avian viruses appears prudent,
Yes.
which I would agree.
The other thing
And
that
excluding bats from contact with farm animals, I think, would be prudent.
That would be
Look
hard
on
to
that
do.
one. Imagine how many people are conducting surveys with mist nets to catch bats, to tag
Yes.
them and release them. How many of them are carriers for influenza viruses? I mean, there's plenty of opportunities for the bats and humans to come in contact with each other as well.
Well, I don't know how many people are actually doing that, Dixon.
I think a lot of people are. I really do.
Are you advocating no sampling, no
Not at
wildlife
all.
sampling?
No, not at all. But it just creates an opportunity for people and bats to come together.
I would hope
And
that the people...
spelunkers.
Yes. Okay. So, yeah, I would hope that the people who are doing the sampling of bats for other scientific purposes are very careful about this sort of thing. I mean, not just because of flu viruses, but because of rabies and because of coronaviruses. I would be much more concerned about people who have less training, who might accidentally contact bats. Spelunkers, you mentioned. Anybody who works in attics or around structures where bats might roost.
Yeah.
But that's a lot of people, to your point.
Indeed.
Yeah.
Indeed.
A lot
Dixon-
of people have contact with
Dixon,
bats.
you said prevent contact between bats and wild animals?
No, I said prevent contact between bats and farm animals.
Farm animals. Well, there you have to keep the animals indoors, basically.
Yes,
But bats can fly indoors, right?
they
You
can.
sure can.
And
They can
that's the
squeeze-
Nipah story, and my concern here would be if these bats are in contact with pigs and you get this virus going into
Yeah.
pigs, then maybe that's the
Yeah.
intermediate host you need.
All right. Okay, that's it. Let's do some email.
Oh, good.
Dixon, last week we did an all-email episode.
I'm sorry I missed it.
Yeah.
No, it was fun. It was me, Alan, and Brienne.
Oh, that is that. I didn't want to step on anybody's toes.
No worries.
You wouldn't have.
Alan, could you take the first one, please?
Sure. Braden writes, "Thank you for reading and answering my question. I'll check out the Cancer Genomics Project to see if there's a way to minimize my risk of dying from pancreatic cancer. I think it was the email after mine that talked about comment threads. The project that I did to spiff up my CV for grad school applications was about motorsport Twitter, basically reading the comment threads and using grounded theory methods to analyze it all. I've also been part of a few esoteric WhatsApp groups in my hometown for the last several years, ostensibly to promote my events. These are hotbeds for RFK Jr. fandom and disinformation. My posts regularly go like, "Sorry to be the science buzzkill, but..." For example, an allopathic doctor is better placed to treat your spider bite than an aromatherapist. While there is peer-reviewed evidence to support that herbal supplements helping cancer patients, ask your oncologist about drug-drug interactions before using it. /ivermectin cures rosacea because that's a parasitic infection, but it isn't effective against COVID, even though I'm sorry for her and her family that Jackie Stone had her medical license revoked for prescribing it. Comments along those lines. I stay in the groups because I've received feedback in private messages that people appreciate someone speaking up for common sense. If your man wants to effect change in people's minds, he can join a similar group and be as civil, compassionate, and calm as possible. Randoms yelling onto internet don't change people's minds. People we know are far more credible than some guy in a lab coat on the news or someone who trolls us on social media. That's why you're more credible to me than RFK Jr., who I've only ever heard stories about. I like to think that during the recent polio outbreak in Harare, some people from the group allowed their kids to get OPV, update vaccines, because I spent 2 days explaining, from knowledge gained on TWiV, the risks and relative risks and benefits of vaccines, and that if they could get IPV from their private doctor, that was the best option, followed by getting OPV from the WHO people who went to the school, and natural polio infection brought up the rear with side effects like lifelong disabling paralysis, living inside an iron lung, and death. I included a list of things like, quote, feed your kid anti-inflammatory foods to keep their immune systems focused on the task at hand, and book an appointment with a Reiki healer for after the vaccine appointment to fill them up with good energy, in the hopes that having a possibly psychosomatic toolkit to minimize ill effects would help them overcome their vaccine hesitancy. Yes, taking my stack of peer-reviewed research about Russian-sponsored anti-vax disinformation around to beat the sense into them would have been more satisfying, but it's possible that a few parents consented to their kids receiving an OPV booster, with a Reiki healing chaser, because they knew me and I could speak their language. Have a great day and keep up the good work, Braden. Cool.
What is Reiki healer? Do you know?
Reiki, I think, is that the one where they place warm stones on the body along chakras or something? And I gather it's a quite innocuous alternative therapy thing that if it makes you feel good, fine.
Uses gentle touch and placement to heal you.
Sure.
Okay. Do you think, Alan, that the Russians sponsor anti-vax disinformation in the U.S.?
Oh, we know that.
We do.
That's been documented that one of the many aspects of the Russian disinformation campaigns on social media was built around sowing discord on issues including vaccines.
Okay. Dixon, can you take the next one, please?
I would be more than happy to. Bryce writes, "Hi, this week in virology team. My interest in virology has been growing while watching your podcast for the past few weeks and while taking a virology course class, BI 115 at Caltech this term. It has been fascinating to learn about the ways the immune system can combat viral infections and the different treatment and vaccination approaches that have been tried and used for fighting different types of viruses. One thing that particularly piqued my interest, discussed during your podcast, was the additional roles of B cells in combating viral infections beyond producing antibodies. I had a couple of questions related to some of the podcast episodes that you have put out over the past few weeks. On TWiV 1103, you talked about two protein substitutions boosting the expression and stability of the spike protein in many coronaviruses. A model came out of Bill Clemens' lab at Caltech called IMPROVE for predicting the expression of integral membrane proteins in E. coli based on their sequences. Are there similar computational models to improve that have been used for boosting the expression of viral envelope proteins like spike proteins discussed? On TWiV 1099, it was mentioned that it is rare to find two identical genomes of hepatitis C virus, even in the same patient. What is it about this virus that allows its genome and the associated proteins to be so tolerant to mutation? Would such a high mutation rate not lead to many dysfunctional viruses being produced? Thank you for all you have taught me through your podcast and for taking the time to look at my questions. Best wishes, Brace.
Hmm.
Very thoughtful and interesting letter from a newbie student of virology. That's pretty deep questions for a person that's just recently been exposed as a.
I don't know about models to boost production of envelope proteins. I'm not aware of any of those. I know that when we talked to Jason McClellan in Texas, he talked about ways to design vaccines, which include, adding prolines, but also adding disulfide bonds and trying to fill in pockets on molecules that lead to conformational change and so forth. I'm not aware. Do you know of any, Alan, models?
No, it hasn't come up, but that doesn't mean much.
Yeah, me too. Jason would know because he might use them.
Yeah.
And the reason Hep C, they're so it's very interesting. The mutation rates for RNA viruses are all pretty similar in terms of the error frequency of the polymerase, with the exception of the coronaviruses, which have error correction. So Hep C is just as error prone as poliovirus. And so if you track poliovirus as it goes from the mouth when you ingest it to the feces, in about 5 days it changes 5% of the genome. And so it varies just as much as hepatitis C. And you ask, would such a high mutation rate lead to many dysfunctional viruses? Yeah, it could, and they would be removed by purifying selection, right? Just like ORF8 null, if it were bad, it would be removed.
Yeah, a lot of the genomes that get produced in any viral infection, but in an RNA viral infection in particular, are going to be broken. And they'll, if they get packaged, they'll get sent out, and that's a particle that won't initiate a productive infection. But the virus is producing so many copies that some of them are going to be fine.
The other part of his question is, what is it about the virus that allows the genome and proteins to be so tolerant? So the protein, that's the key. Many viral proteins are more tolerant than others, like the spike of SARS-CoV-2 can tolerate many amino acid changes, right? But what is it? The spike of measles virus is less tolerant. We did that study, remember, which we likened to the airplanes that came back with the bullet holes, right? They reinforced those parts of the wing. So measles cannot sustain changes. It must be a structural thing, right? All right, Terry writes, your videos not only got me through my own virology course at University of Alaska Fairbanks, but you have inspired me to go into virology and eventually work in biosecurity. I'm actually presenting as an undergraduate at the AAAS Regional Conference in San Diego this June on the boreal pox virus, formerly known as Alaska pox. I cannot thank you enough for the inspiration and giving me the courage to follow through. You are one of the authors of my textbook that I cherish, and I just needed to say thank you for everything. Thank you, Terry. We appreciate you. Thanks. So many thanks. We do love hearing stories of how we help you. I think we're back at the top with Alan, right?
Back to me. Yeah. Anthony writes, the cartoon image attachment in the preceding email was posted on Facebook as an example of the idiocy/lunacy of anti-vaxxers. I thought it too ridiculous to be so, but I found in the Chicago Tribune article that the image had been posted by one of the Trump delegates. This is a cartoon of cattle waiting at the slaughterhouse, and one of them is complaining that one of the ones ahead of him is not wearing his mask. Implication obviously being that taking SARS-CoV-2 seriously is like being led to slaughter. So, yeah, that's pretty insane. Cartoon was sent to me by a Hudson County resident and anti-vaxxer. I was shocked that anyone would think this image to be insightful or persuasive. If the individual had recently arrived from some backwater or was intellectually challenged, I'd not have given it a thought. But this person grew up in Queens, New York, and had worked in China for a major U.S. financial publisher. Remember Ionesco's rhinoceros? Ionesco's rhinoceros?
I'm looking it up.
Yeah.
It's a play by Eugene Ionesco.
Right.
Oh, it's the Theater of the Absurd.
Indeed.
Do you know that, Dixon?
I know the Theater of the Absurd. I didn't realize that the particular episode Rhinoceros was in it. But,
Over the course of 3 acts, the inhabitants of a small French town turn into rhinoceroses. Ultimately, the only human who does not succumb to this mass metamorphosis is the central character. Blah, blah, blah. The play is often read as a response and criticism to the sudden upsurge of fascism and Nazism during the events preceding World War II, and explores themes of conformity, culture, fascism, responsibility, logic, mass movements, mob mentality, philosophy, and morality. Okay? That's Ionesco's Rhinoceros.
Thank you, Anthony, for that reference. I'm sorry I didn't get it.
The real reason he didn't convert to rhinoceros was because he didn't want to horn in.
Right. By the way, there seems to be considerable intersection between anti-vax and hatred of cars.
That's interesting.
The electric car mania, I strongly suspect to be a facet of the Oedipus complex. Removal of the internal combustion engine is equated with castration, and perhaps EV sounds like ED.
Gosh.
Okay. Hatred of electric cars, maybe, is what you meant?
Yeah, I think so.
Okay. Well, electric cars have more thrust, so take of that what you like.
Oh my gosh.
Sorry, it's a family show.
Dixon, you're next.
I know. I'm not sure I can go on. John writes, Vincent and the TWiV team. Based on the comments of a few TWiV episodes ago, when H5N1 was repeatedly mentioned in the news, I thought I would send along a new map with data updated to April 20th, 2024 of the confirmed cases of wild birds infected with avian H5N1 influenza. The map contains more than 10,000 data points and gives the location at the county health department reporting level, the species of bird infected, and the strain of H5N1. Oh, that's interesting. I sent in another map to you almost 6 months ago, which you talked about on the air, but this map is now complete and accounts for the extremely rapid increase in case reporting. And then he gives a link to that. Also, we have now compiled the data on mammals from the USDA Plant and Animal Health Inspection Service, whose data now confirms more than 200 domestic and wild mammals that have died from avian H5N1. Here is the map for that, if you're interested. And he gives the next one. I love the show. That is an understatement, as there is nothing else like it out there, and I've been listening and supporting since the beginning. You should have a GIS scientist on one of those days to talk about the difficulties in mapping the spread of things like H5N1. That's a great idea, which is a nightmare map to map and keep track of at the field level. You know, there's no question about this. And the best thing I've seen so far for something like this has been the epidemic of opioid overdose. I'll send you the map of that. It's not a map, actually. It's a single figure, but the width of the figure at the particular time of the year equals the number of deaths from opioid overdose. And it's alarming, to say the least, to see it visualized like that, because you can envision how this thing works, and it's still out there, and it's still having its effects. I don't know what to suggest about this, except the more easy it is to see what's happening, the more understanding there will be.
The maps are amazing.
Yeah. Yeah, and I think this is a great idea if we can get an epidemiology GIS person on.
I would love that.
John, if you know someone.
Or if you are someone. John is a lecturer in applied mathematics and geographic information science, so this graphic is very small.
That's
Johns
GIS
Hopkins.
is, right?
Yeah. So geographic information science is GIS.
Actually, I know someone over at Hunter College, if you're interested. His name is Sean Ahern, and we actually published a paper once on the West Nile virus epidemic as it was evolving, and he's really very good at it.
So the first map, which is the H5N1 isolations, you click on each dot and you get information about where
That's
it's from.
just the way it should be.
Oh my
That's
gosh.
terrific.
Ohio, Bald Eagle, February 28th, 7 p.m. Wow. And then it's all over the U.S. Look, except some places are sparser than others.
But that may just be a population issue.
Yeah. And then there's one of mammals, which is far less, but let's click on the one in New York. And that is a red fox in Rhode Island.
Huh.
This is great. We will put links to this. You can have fun.
This is cool.
With this. The mammals seem to be north biased, don't they?
Maybe, yeah.
Maybe they're not looking in the south. I don't know. Thank you, John. That's very cool. All right. We have another email, but we'll save it for Angela because it's a vet question.
Right.
Which now brings us to our picks. And Dixon, what do you have for us?
Well, I have two of them. The second one was a short pick, but it's really worth watching, however. The first one is a video produced by NASA on a theoretical journey that you could take to the Andromeda Galaxy. And there has been so much detail revealed now that the James Webb Space Telescope is in operation that you can actually go to various parts of the Andromeda Nebula and see what it would look like if you turned around and looked back to the other way and saw what our galaxy looks like from the Andromeda Nebula. Well, the significance of all that is that the Andromeda Nebula is slowly but surely moving towards our galaxy. And
that's the Andromeda Galaxy.
That's the
nebula.
Andromeda Galaxy. Did I say? What did I say?
You said nebula, but it's
I'm sorry.
It's all right.
I meant galaxy. The thing that I was confused about in the beginning of all of this a long time ago was that why are some things moving towards each other when Hubble says everything is moving away from each other? Well, that's true on a large scale, but on a small scale, you have these perturbations. And so in about 4.5 billion years from now, and I'm sure everybody's out there biting their fingernails worried about all this, these two galaxies will join and become one with one another, as it were. And as I put it, we will become scrambled eggs because the Andromeda Galaxy is more organized and and probably is heading towards us more than we're heading toward it. But it's amazing to think about the galactic pressures that are at work for the gravity to be so immense to include trillions of stars from each of these two galaxies. I can't possibly imagine it, but you can watch it actually happen before your eyes. So I thought that was a startling thing to think about in the long term. But in the short term, I thought this snippet of the highest jump ever recorded by a human being, and it was documented by this guy jumps like 50. I forget how high it is.
He hits a 12.5 foot high.
He's at the top of the scale. You can't go higher than that because they don't have a way to measure it.
Well, a basketball hoop is 10 feet, and he's grabbing 2 feet
He could hit his
that.
head on the rim.
Yeah.
The thing is that you wonder about things like this, whether they're actually photoshopped or faked or something else. And I don't think so. I've actually seen this where he was doing several practice jumps first, and they were all equally off the charts. So that's just for an enjoyment's sake.
Very cool.
Those are my two picks.
So we had some corrections from last week for you about collisions and black holes.
What did I say?
We read some letters about.
Oh, great! You mean I got roasted? And I
correcting
wasn't even
your
there
astronomy.
For it? I couldn't
no,
defend
you
myself?
weren't exactly roasted. It was astronomers writing in too.
Oh dear! Oh.
But they said you got most of it right. They were just
a
few
few things.
Right.
I got most of it right.
They said they love you anyway.
Yes, you're still a star to them.
A fading star, no less. But I'm running out of hydrogen, for God's sakes.
Alan, what do you have for us?
I have one pick, but two links. One is a gallery that Nature put together about what a scientist looks like. And I love this thing. It's a bunch of pictures. They got clearly they got professional photographers, or at least people who could be professional photographers, to do these portraits. They're very well shot, and I love the diversity that it shows, not just in the appearances of the people, which there's a lot of, but also in the activities in which they're engaging. So it's not a bunch of people standing around in lab coats. Some of them are, but it's not. It's also not just field scientists standing with a mountain in the background, though some of them are. It's just all kinds of neat stuff that people are doing that is scientific research. You've got an astronomer looking into a huge telescope. You've got somebody in a greenhouse, and somebody handling a snake, and somebody, in a rainforest, and someone in the lab. And
I love it.
It's just really, it's a great, I think it's just a really good public outreach effort for people to get their minds around what science really entails. And then they took this all a step further. They have put 50 of these photographs on public display at bus stops in London.
Oh, cool.
Like where you would normally see an ad, there's one of these pictures of one of these scientists explaining what this person does. And I think that's really cool, and would love to see them do that in more cities. I know they have offices. Right. Nature has offices in New York, so that seems like it ought to be something they do.
Right.
Maybe elsewhere too.
Wow.
Neat.
Yeah.
Well, scientists look like everyone else, basically.
They look like everybody else.
They do.
They're doing all kinds of cool things.
They are everybody else.
Yep.
All right. I have two picks which relate to what's been happening this week at Columbia, which is there have been protests, of course, and on campus, which brings you back to the 1960s. Dix and I were talking about Mark Rudd and Grayson Kirk and the Students for a Democratic Society.
The SDS, so they're sodium dodecyl sulfate.
Yeah, that's the way I always thought about it. That's right. That's right. Anyway, there's one article in the Atlantic. They're just views on the protests, which I think are very good. The campus left occupation that broke higher education by George Packer. Elite colleges are now reaping the consequences of promoting a pedagogy that trashed the post-war ideal of the liberal university. So basically, liberal universities are supposed to be places where you can say anything you want, right?
Well,
where else in the world can you do that? You can't protest at many other places. And so now we see, reactions to that. He says they started all in the 1960s with the occupation of the president's office at Columbia University.
So basically it's the fault of the boomers.
Is it?
I don't know whose fault it is, but...
That's how I see it.
Well, last night they said the largest protest ever on campus was after the news leaked out that Nixon had ordered the bombing of Cambodia. And McNamara said, we have not done that. And then Nixon admits that we have done it. And that triggered an enormous response from students. The most that ever, like 900 campuses, including high schools and grade schools, everything. They just went crazy. I don't remember that event, actually.
Well, you notice that the protests are not just at Columbia. They're at many universities.
There are many places.
Right,
because those are places where you can. If you think about it, you can't just go out on the street. You need a permit, and if you don't have one, you're going to get arrested. You can't do it in a building of a business. They're not going to let you do that. So that leaves campuses. And now this article is arguing, now they're cracking down against the thing that they think they're promoting. So that's kind of an interesting dichotomy.
Yeah.
Anyway, so that's interesting to read. And then my second one is an opinion by John McWhorter, who's a Columbia professor. It's called, "I'm a Columbia Professor. The protests on my campus are not justice." And, there's a photograph. So when I went last Wednesday, over a week ago, right, the main gate was closed. There were police with riot helmets there. There was a line of people protesting outside. And then inside there was some kind of demonstration. And we had to go around to to get in another gate, but that's all fine. And then on Monday, they said, don't come at all, teach remotely by Zoom. And then we went back this following Wednesday. I think this is a good perspective because he's arguing basically it's not peaceful, and you're entitled to protest, but you're not entitled to be anti-Semitic, for example. That's not what protest is about. But he also says that if these people had been chanting anti-Black slogans, they would have been shouted down immediately by other students and so forth. But I think they're both interesting perspectives, right? So that's why people have sent them to me, actually, in the course of the week. So I thought maybe people would be interested in what's going on. I don't know what the solution is when, the university itself cannot make the changes that they want, right? Columbia cannot end the conflict.
No.
Right.
Someone else, but they were going to stay there at Columbia until, it doesn't seem to be rational to me, but I don't know. Anyway, they're interesting.
No, I can understand protesting for the university to divest investments from particular things because that is something that is in the university's power.
Yeah.
But saying, we're going to protest until there's peace in the Middle East, that's not really something
so in
the
the
dean
60,
can fix.
In the 60s occupation, the demand was to Columbia stop investing in military research.
Right.
When I was in college, there was a whole raft of protests around divestment from South Africa.
Exactly.
That's
Right.
And divestment from companies that had significant presence in South Africa. And, that's the kind of thing that I think makes sense. Whether you agree with the cause or not, but that's a kind of demand that you could make in this context, and it would be reasonable.
Well, he mentions that in this article as well, that the South African protests. Yeah. All right. We also have a listener pick from David. Back in the 90s, one of the highlights of my video career was being able to record the U.S. Navy's Blue Angels at an air show in Kalamazoo, Michigan. But the ground-based footage I got is nothing compared to the visuals captured by the camera affixed to the bottom of the team's lead plane seen in this YouTube video. Containing what amounts to their entire show over San Francisco in October 2019, it is both a thrilling demonstration of the Blue Angels' extraordinary flying skills, as well as a virtual overhead travelogue of San Francisco region, including such landmarks as the Golden Gate Bridge and Alcatraz. And while the visuals are thrilling, the soundtrack, which is muted engine noise, is paradoxically soothing, being essentially a form of white noise. If you don't have time to watch the entire 45-minute program, at least the last 15 minutes contain some of the best bits. And he gives a YouTube link.
It is a very cool video.
David is Microbe TV's video editor.
Oh, wow.
Who will be hearing this read as he's editing this video on Saturday sometime. Thank you, David.
Fabulous.
Yeah.
That's it for TWiV 1109. Show notes are at microbe.tv/TWiV. If you want to communicate with us for any reason, a pick, a comment, a question, the email is twiv@microbe.tv. And we would love to have your financial support. It means that you enjoy what we do, and it also will help us do more of it. You go to microbe.tv/contribute. Dixon De Pommier is at trichinella.org and thelivingriver.org. Thank you, Dixon.
Thank you, Vincent, and thank you,
Not Daniel. It's not Daniel.
No,
Whoever I am.
Alan the Dove. Yeah, and how dare you speak out against war?
Yeah, that's a very good way to say it. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.